Dear students, the last few classes we were talking about the applications of biotechnology in various fields. We were talking about its role in medicine, in gene therapy, stem cell therapy, etc. Now the fourth application of this biotechnology, that is molecular diagnosis. Normally we are diagnosing the disease by the presence of the bacteria, viruses or any other pathogens. But the early diagnosis cannot be made with these microorganisms. So you can just find out the disease only after getting the symptoms produced by the pathogens. It will take some time, and also by the time we have more concentration of pathogens in the body. To avoid this one, now a new technique has been adapted to diagnose the disease at the earliest time. That is called molecular diagnosis. So, early diagnosis of various infectious diseases or genetic defects normally is essential or needed for appropriate treatment. We have a number of conventional methods and normal methods. The conventional methods are not actually reliable, not used to find out what we have the early diagnosis of the disease. We have some of the conventional methods, the traditional methods. For example, microscopic examination, urinalysis, urine analysis. Then we have also the serum analysis, etc. So, early diagnosis is not possible with these conventional methods. That's why now the scientists are searching for a specific, a sensitive and a simple diagnostic techniques for diagnosing the diseases. They have found a number of the methods. 3 methods mentioned in the book. I mentioned about the conventional methods. So these are all the laboratory techniques. These laboratory techniques are indirect methods. They are not always easy that one specific, not giving accurate measures, accurate values. So that's why normally scientists are continuously searching for a specific, sensitive and simple diagnostic techniques for the diagnosis of the diseases. Okay, and some of the methods are, you see, PCR, polymerase chain reaction. Number 2, ELISA, for detecting what we have HIV in the body. And also, the most important one, recombinant DNA technology. So, recombinant DNA technology, then what we have PCR, polymerase chain reaction method, and also we have the ELISA method, that is a test to find out the early just presence of the organisms in the body. Okay, that's about the introductory part. Now I mentioned about there are 3 different methods which are highly reliable, used or help in diagnosis of the disease. Now what are the 3 methods now? Other than the conventional methods, some of the reliable methods, techniques that help in the early diagnosis are, number 1, recombinant DNA technology. Number 2, polymerase chain reaction. We will discuss later after this one. This is concerned with the amplification of genes. We can increase the number of genes or multiplication of genes in billions. That is normally with the help of polymerase chain reaction, concerned with the amplification, increase in number. Then ELISA test, the one which is used to find out whether the person is HIV positive or not. Early diagnosis. So all such that is what we have, the methods or techniques are concerned with molecular diagnosis. Diagnosis at the molecular level, not by means of conventional methods. So these are all more reliable and help the early diagnosis of the disease. Now how can you know the presence of bacteria and then viruses in the body at the earliest? So normally you can get the idea, the presence of the pathogens like bacteria and viruses only when these pathogens produce symptoms. After the appearance of the symptoms only we came to know that one we have affected with the disease. So this is now the early diagnosis. But we have actually before the symptoms appear. Early diagnosis can be done even in low concentration of bacteria and viruses present in the body. So with a low concentration of bacteria and viruses in the body, even before the symptoms appear, early diagnosis can be detected by amplification of their nucleic acids, increasing the number of nucleic acids by means of PCR, so that we can find out the presence of bacteria and viruses in the body, even in low concentration, even before the symptoms actually do not appear. Even before the symptoms do not appear, we can find out the presence of bacteria and viruses. There is a method, there is the importance of what we call the molecular diagnosis. There is a very important method. Also, there is that its importance is diagnosing the disease at the earliest time, even before the symptoms appear, even in low concentration of viruses and bacteria present in the body. Now in this syllabus, we have the test, ELISA test is given in detail. After that we have to go for polymerase chain reaction. Now let's take what is ELISA. Under molecular diagnosis, we have to study first one, that is what is called ELISA. The expansion for that one, enzyme linked sorbent assay. Enzyme linked means here the antibody is linked with an enzyme which will react with a substrate to produce a colored reaction. That's why it's called enzyme linked. Antibody linked with an enzyme that creates a color reaction, that creates what is called a color product. That is why it's called enzyme linked. We'll talk about. Then sorbent. This is nothing but adsorbent. Adsorbent or sorbent. That is, normally in this case, in this test, either the antigen or antibody can be detected, which can be normally just bound to a solid support, what is called the plate, normally called as ELISA plate, micro titer plate, made of plastic, polyvinyl chloride, like that. So here it is called a sorbent assay, using a plastic plate, what is called polyvinyl chloride, and which is forming the base, what is called assay plate, on which only the test is carried out. Hence called actually sorbent, adsorbent. So the antibodies or antigens are adsorbed onto the plate, ELISA plate, while doing the experiment. Hence called sorbent. That's the meaning for sorbent, adsorbed, bound to the surface. Assay means we have a series of steps to find out the result. That's called assay. That's why the name is given as enzyme linked sorbent assay. Enzyme is linked to the antibody, forming a conjugate, secondary antibody, and that one, or we can say the detection antibody, that's the one which will decide whether the antigen is present or not. Okay. Now what is ELISA? It's a biochemical procedure, normally, to detect the presence of either antigen or antibody present in samples. For example, just we see in a sample of either the blood serum or the urine, etc. In urine, they are finding out what is called human chorionic gonadotropins. This is one of the important hormones nowadays used to detect whether the woman is pregnant or not. The presence of that hormone indicates the pregnancy. So, either from the serum or from the urine, either the antigen or antibodies are actually tested to give the result. Now, ELISA was first discovered by two people in 1971. So, Eva Engwall, or Engwall, we can say English, Engwall, and Peter Perlman in 1971, what I mentioned. Now, what is the importance of or significance of this ELISA? So, it is a very important diagnostic tool to determine if a particular person is HIV positive or negative. This is number one. Secondly, it is a tool to determine the serum antibody concentration, the one which is produced because of the entry of pathogen. For example, antibodies produced because of the infection with the pathogen, the one which produces antibody, as in the case of HIV. That is a one. So you can find out the serum concentration of antibodies, or you detect the presence of antigens for human chorionic gonadotropins. Human chorionic gonadotropins, simply called as HCH, human chorionic gonadotropins. This is not the growth hormone, this is human chorionic gonadotropins. This hormone is released in the case of pregnant women along with urine. This is a urine analysis. If the hormone is present in the urine, that indicates the woman is pregnant. That is a test. So this is a one. Antigen present, actually detecting the presence of antigen in human human chorionic gonadotropin, a hormone present only in women during the pregnancy, released by the placenta. It is released along with urine. That indicates the woman is pregnant. That is about what we have, the important, two importance, to find out whether the person is positive or negative to HIV, and also the second one, to find out the concentration of antibodies in the case of infected individuals, either by HIV or anything, or to detect the presence of antigens for human chorionic gonadotropin, a hormone that is released along with urine in pregnant women. So it is a test for pregnancy. That's about the importance. Now let's see the procedure. Another importance of ELISA test is, it is a tool to determine the serum antibody concentration. How much antibodies are present after the infection with the pathogen. For example, when a person is infected with the HIV, it stimulates the production of antibodies. So one can determine the amount of antibodies present in the plasma, or we can say the concentration of antibodies in the serum. That's called serum antibody concentration. So that because the virus is acting as an antigen, stimulating the production of antibodies. More the amount of antibodies, more is the infection. That is one of the, actually, that is a result we can get it. Then, or we can detect the presence of antigen for certain hormones. In the book it is given to detect the presence of antigen and hormones. It is not and hormones, it is used to detect the presence of antigens for hormones like chorionic gonadotropin, human chorionic gonadotropin. This is a hormone secreted by placenta. This is a hormone secreted by placenta during pregnancy. So only during pregnancy this hormone is secreted, because the placenta is found only during pregnancy. So the presence of chorionic gonadotropin in urine is an indicator to confirm that the woman is in pregnant state. So it is an indicator of pregnancy, because it is secreted only by the placenta. The placenta is found only during pregnancy. Hence the presence of human chorionic gonadotropin is an indicator of pregnancy. It is normally released along with urine. To test the urine by using this molecular analysis, we can find out whether the woman is pregnant or not. Okay, that's about, it's accurate also. It's about the importance. Now we have to go for the mechanism of ELISA test. What are the actually steps involved? Okay. Let's see what are the steps involved during ELISA. Now it's an assay test. I mentioned already the word sorbent, referring to the binding of either antigen or antibody to a plate. That plate is called what is known as ELISA plate. It's normally plastic in nature, made up of polyvinyl chloride. So that plate is also called micro-titer plate. That plate is also called micro-titer plate or assay plate. Micro-titer plate or assay plate. Okay, now, so you are having what is called the wells. The word wells refer to nothing but what we have the tubes, the test tubes or the vials found in ELISA kit. It consists of a number of just the test tubes, and such a test tubes are called the wells. Okay now. So what we are doing that one, during diagnosis, the sample suspected to contain antigen. So I am describing only what is called the direct ELISA, because there are 4 types of ELISA: direct ELISA, indirect ELISA, competitive ELISA and sandwich ELISA. I will explain separately. Here I am talking about the procedure adapted in the book, the direct ELISA. So during diagnosis, the sample suspected to contain antigen is immobilized onto the wells. Immobilized means attached, not able to move. It is being stuck onto the surface of a well, a coated well. So it's being added not directly the antigen, it is being used, added to the what is called the wells along with a buffer solution. And after adding, the buffer has to be removed and the plate is washed like that. So during diagnosis, the sample suspected to contain antigen is immobilized, that is found on the plate without allowed to move. That's called immobilization onto the wells on the surface of ELISA plate. So the ELISA plate is formed of polyvinyl chloride. It's also called assay plate or micro-tighted plate, made of plastic simply, on the surface of which only we are making the antigen to stick. So the antigen is allowed to stick on the surface of this plate. So it is found in a what is called a well with a buffer solution. Now and then we have to remove the buffer solution and wash it. And finally you have to find out. It is a colored reaction only. So it's a biochemical procedure, not we are finding out what we call the organism, just like HIV, it is impossible. That's why we are finding out the antibodies produced or the antigen of that virus. Okay, so I am just explaining only direct ELISA. Now the antibody specific to this antigen means the antibody, the one which can react with this antigen, is added and allowed to react with the immobilized antigen. The antigen is immobilized. Now we are adding a buffer containing the antibody. Now the antibody is allowed to react with that antigen, and such antibody only just added to the what is called the well. Here the well referring to, that's what's called the chamber, or we have the glass vessel, or we have test tube found in what is called ELISA kit. Now then the antibody, the one now after reaction with what is called antigen is called anti-antibody. Now this anti-antibody is linked to an appropriate enzyme. We have different types of enzyme. That's why it's called enzyme linked procedure, enzyme linked assay. Enzymes such as peroxidase, the one which catalyzes hydrogen peroxide. That enzyme is called peroxidase, which catalyzes the substrate hydrogen peroxide. Now after the treatment with the after this antibody with the enzyme linked is added, so that is what is called actually antibody linked. Just we can say secondary antibody, enzyme linked conjugated antibody or detection antibody we can say. That antibody with the enzyme is called detection antibody. Okay, now we have the detection antibody with the enzyme peroxidase. So after that enzyme is added, we have some more antibodies present which are not bound with antigens. They are called unreacted antibodies. So the unreacted antibodies are removed and washed away, including the buffer. So the unreacted antibodies, those antibodies which will not react with antigen present there. So only few antigens, these few antigens react with the antibodies. The remaining antibodies, they are called as free antibodies, not reacting with that one antigen. They are called untreated antibodies. They should be removed or washed away. They should be removed or washed away. So then to this actually what is called the well, we have to add a specific substrate through a buffer solution. The substrate is normally the one which will react with the enzyme. So that substrate normally is that one, a substrate specific to this enzyme. So the enzyme is peroxidase, which will normally break down hydrogen peroxide. So we have to add the substrate in the form of hydrogen peroxide. along with some color agents, because the intensity of antigen can be just counted only by the amount of color produced. More the color, then more the intensity of color, then the amount of antigen present in the sample is more. So, hydrogen peroxide is substrate along with some color reagents. The color reagents may be in the form of 4-chloro, that is naphthol, which can produce a color. That is called as a color agent. So, first you have to add the antigen that should be immobilized in the wells using the buffer. Then we have to add the antibody which will react with that antigen. Then we have to add another antibody which is linked with an enzyme, a specific enzyme, here what is called peroxidase. Then we have to add the substrate after removing the extra antibodies which are not reacted with antigen. We have to add the substrate, the one which is being catalyzed by the enzyme peroxidase, and that is nothing but hydrogen peroxide. To get the color in that solution, that is in that buffer, we have to add what is called, that is a color reagent. That color reagent is nothing but 4-chloronaphthol. Okay. The substrate is catalyzed by the enzyme, here peroxidase, resulting in generation of color. Now the intensity of color is normally read out by spectrophotometer or ELISA reader. There is a ELISA reader or spectrophotometer by which one can find out the intensity of color. So the intensity of color is directly proportional to the amount of antigen present in the sample. If the intensity of color is more, that means more antigen is present. If the intensity of color is less, that means less antigen is present in the given sample. That's about the procedure, and this is meant for what's called direct ELISA. As I mentioned earlier, there are 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and finally competitive ELISA. In the book, what is described is nothing but the direct ELISA. I will give the diagram also after finishing this one. Before going to that one, what is the importance or significance of ELISA? The ELISA test is highly sensitive and detect antigen that is present in the sample even in the range of nanogram. That means even the smallest amount of antigen present in the sample can be detected by this method. So it is specific and highly sensitive method for diagnosis. That is the second one. And also it doesn't require what is called radioactive isotopes or radioisotopes, and also doesn't require radiation counting apparatus. That means we are not using radioactive isotopes here or radiation, just what we have apparatus for counting. So ELISA normally possesses the added advantage of not requiring radioisotopes and radiation counting apparatus. That means we are not using radioisotopes, so there is no need to have what is called radiation counting apparatus. That is about the significance. I will represent the diagram for the direct ELISA, what is given, and then I will represent a diagram only, that is what we have indirect. In the book also given sandwich ELISA, and also we have competitive ELISA, only the diagrams. Okay. The following illustrations show the various steps involved in ELISA test. What I represented is nothing but the direct ELISA test. Now, step 1. Now this is a vessel. Adsorption of antigen onto the wells in ELISA plate. Now at the base we have the ELISA plate, otherwise called microtiter plate or polyvinyl chloride plate. In coating buffer, ELISA plate in coating buffer, this is a buffer solution. With the buffer only we can add this antigen, not directly add the antigen to the well. Then after that one, we have to remove buffer and wash plate. Removing the buffer and washing the plate and placing the plate once again. Step 2, blocking. This is coating, now this is blocking. Now here the antigens are added and immobilized on the walls or to the wells. Okay, now blocking. Buffer with unrelated proteins is used to block the free reaction sites. So other than this area, these are all the free reaction sites, they should be normally blocked. Otherwise some reactions are taking place. Once again you have to remove buffer and wash plate. Step 3, detection. Enzyme conjugated detection antibody. Now this is what is called enzyme linked or conjugated detection antibody. It is normally added and it binds with the antigen. Then step 4, readout. Substrate catalyzed by the enzyme. We have the substrate catalyzed by the enzyme giving the product generates a color that is being actually read out, and that is called readout. Substrate catalyzed by the enzyme generates colored product, or we can say colored readout. Colored readout. So these are all the steps. By using the spectrophotometer we can find out the intensity of color. I mentioned more the intensity of color, more the antigens. So this is a simple mechanism of direct ELISA. The meaning for direct ELISA, the antibody directly contacts with the antigen on one side, on the other side with the enzyme, and generating the colored product without others' help or without another antibody's help. That's why it's called direct ELISA. Directly contact with the antigen and enzyme and produces a color. Types of ELISA. What we have learned is nothing but the direct ELISA. There are 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and then competitive ELISA. In direct ELISA, the enzyme linked detection antibody directly conjugates with the antigen and detecting the antigen. In the case of indirect ELISA, there are 2 antibodies. One antibody is called primary antibody, which conjugates are linked with what is called antigen. Another antibody, what is called conjugated secondary antibody, linked with enzyme. So here just actually indirectly we are finding out the colored product, that is substrate and product. So this is directly conjugated primary antibody, means primary antibody is the one which is always binds with antigen. Secondary antibody is the one which is always binds with the enzyme. Here 2 antibodies are involved, that's why it's called indirect ELISA. Now in the case of sandwich ELISA, now here actually what's happening, there is one antibody which is normally immobilized and attached to what is called the plate, assay plate or ELISA plate. This is called capture antibody. The sample with antigen is added into the medium or added into the well. There you see that when the antigen is captured by this antibody, the antigen is captured by antibody, then the antigen is now binds with another antibody which is called enzyme linked detection antibody, because this is antibody with enzyme. Now here the antigen is sandwiched between what is called the capture antibody and the enzyme linked detection antibody. It's in the form of sandwich. Capture antibody, enzyme linked detection antibody, in between which we have the antigen is in the form of sandwich. Now the enzyme in the detection antibody catalyzes the substrate to generate what is called the colored product, and that's why it's called sandwich, because of the presence of antigen between 2, that's antibodies. The last one, competitive ELISA, here the number of antigens in the sample is more, so that more the antigens in the sample, then fewer antibodies will be able to bind with the antigens. There is a competition between the antigens to bind with what is called antibodies, because we have fewer number of antibodies when compared to the antigens. Hence called competitive ELISA. Which antigen has to bind with the antibody? There is competition among the antigens, because more antigens in the sample when compared to the antibodies which are less in number or few in number. So there is a competition between what is called actually the antibodies. Hence called competition. And the ELISA type is called competitive ELISA. So these are all the different types. With that I concluded with the ELISA. We have to pass on to the PCR, the next one in the next class. Okay, thank you.